good morning, everybody. So I'm Michael Kassiou, and what I want to do is give you a very short description of the potential use of the total body PET, particularly when it relates to drug discovery. And the research opportunities were presented in the previous seminars and again depicted here, but it's really the context of drug discovery and the use of total body PET that I want to run through today. So why is total body PET and drug discovery important? How can PET be useful in that context? So if you look at some of these current trends, particularly in relation to novel drug targets, which is shown in novel drug entities or molecular compounds shown here, over the years, the number of compounds that have progressed to clinical being successful have actually decreased, where the development times, the cost of R&D and sales have actually increased. So drug discovery in itself is very challenging. And on the right-hand side here, you can kind of see that you need to start with a lot of compounds that takes a very long process to go through to be able to get one compound approved, and very costly over a long period of time. So there is an issue with the way drugs are actually being developed and discovered. And when a set of pharmaceutical executives were actually asked the question over the next 3 years, which area will play the most important role to your company in developing the pipeline of new drugs, and molecular imaging was up on top. So imaging has a major part to play in improving the drug discovery process. And why is that? It is because PET imaging itself is translational. So you can use PET imaging or use the technology to evaluate drugs all the way from laboratory animals all the way through to the clinic. So it's a technology that is actually very translatable and further enhanced with the capability of the total body PET. So if you look at the drug discovery process, you can actually see where PET imaging can actually be used here, which is at the discovery stage, but you can also use it in the development stage when you're selecting patients, performing clinical trials, and are interested in evaluating the efficacy of new drug candidates. But developing a PET tracer to use in any drug study is not that simple, and you'll hear from Giancarlo and John as well in more detail around that. But to take a molecule and develop it into a PET tracer, it needs to meet certain criteria. And this is not to be extensive over here, but there are chemistry criteria in terms of the physicochemical properties, the radionuclides, where you introduce the label and when you introduce it, the affinity of the tracer for its target, the type of binding, whether it's reversible or not, but also all the physicochemical properties that are important for validating a radiotracer. So it's not a simple process to actually develop a radiotracer that's going to be useful. But in drug studies, there are essentially three types of experiments that you can do that are going to be very useful, particularly with the use of the total body PET. And the first one is being able to measure the pharmacokinetics of a drug of action, particularly for candidates that have been developed as therapeutic agents. And over here is an example of a couple of therapeutic drugs. There's a lot of kinase inhibitors shown here with these four labeled with both fluorine-18 and carbon-11, protein pump inhibitor, and also an SSRI. So you can actually, as long as they meet the criteria, you can actually take those or candidates that you're developing as therapeutic drugs and radiolabel them and be able to perform pharmacokinetic experiments that tell you something about the molecules in vivo. The more interesting or more exciting part of drug discovery and the use of PET is the ability to measure the pharmacodynamics of a compound. And that actually means not radiolabeling the drug candidate itself, but using some other tracer that actually labels the site that you're interested in to be able to give you some information about the drug that you're developing as a therapeutic agent. And here's an example here where there's an experimental drug here with Johnson we're developing as an antipsychotic. And their experimental drug, the question they ask themselves is, does this drug actually bind to D2 receptors in vivo, in human? And what is the oral dose that they needed to have a receptor occupancy measurement of 65 to 80%? And so you can do this by taking other tracers that are known to be D2 binding ligands, and in this case, raclopride, and you can take raclopride, and this is how raclopride is actually labeled. So it's a very different chemotype, a different molecule to the drug of interest. And here's their study design. And you can see in these baseline experiments with raclopride, you see very nice uptake in the caudate putamen, which is what you would expect for a D2 binding ligand. And then when they administered their therapeutic drug, both at 5 milligrams and 20 milligrams per kilo into the patient, you can see that what they represent is 30 and 65% occupancy. So they get information around the pharmacodynamics, that is, does their compound actually interact with the D2 receptors? And it does. But to get a dose receptor occupancy, you need to do a little more than just 2 time points. And they did this by looking at different concentrations of the drug and different concentration of the drug in plasma. And it has been known for some time that for a lot of antipsychotics, you only need around 60% or no more than 80% occupancy before you start getting extrapyramidal side effects. So they want to determine what dose of their drug correlated with 80% occupancy to give you that sweet spot in terms of therapeutic efficacy. And they did this in looking at the plasma concentration of their drug and correlating that. So they found that a plasma concentration of around 30 to 60 nanograms per mil correlated with 60% and 80% D2 occupancy, which correlated to a dose between 20 and 35 milligrams oral dose. So that those pharmacodynamic experiments are very useful in telling you whether your drug target interacts with the target that you're interested in, but also able to optimize the dose for the individual to avoid extrapyramidal side effects in this case, but any other adverse kind of effects. Here is another example that's kind of more in the early stages of drug discovery. So you can do the same type of receptor occupancy study with 3 different types of molecules. So your chemotype A, B, and C represent structurally diverse compounds that bind to the same therapeutic target that you're interested in. And again, you can do these competition studies or the receptor occupancy studies where these imaging experiments in PET represent no occupancy and then some level of occupancy. And this is shown, in this case, the optimum occupancy for a drug to be useful was shown to be just above 80%. So you can use these 3 different, diversely structurally different drug lead compounds. And the idea here was to determine which one of these 3 would be the best scaffold to progress. And you can see when you do these dose response or receptor occupancy studies, that chemotype 3 gets to the level of the occupancy with the lowest plasma concentration, where chemotype A needs the most. So from a chemistry point of view, a medicinal chemistry point of view, or early drug discovery point of view, it is quite clear that chemotype C is the one that you would progress over chemotype B or A. So again, not just able to tell you something about your drug and the level of occupancy you need for clinical efficacy, but also using receptor occupancy to tell you something about which molecular scaffold is the best one to develop your drug, or use to develop your drug. The third part that you can use PET imaging for, and particularly whole body PET, is for being able to measure the efficacy of any newly developed drug. And that's really, really important because you can quickly weed out whether something progresses or not. And so here's an example using rituximab, which is an anti-CD20, a lymphocyte antigen monoclonal antibody. And CD20 is highly expressed in a number of tumors. And so part of not all patients, but some of these patients, or some cancer patients, do possess or express a high level of CD20. And so knowing which patients to select for that particular treatment, or rituximab treatment, is really important. You don't want to give this antibody to people that don't have elevated levels of CD20. So what you can do here is take the antibody and you can label it here, not using F18 or C11, but a chelator with zirconium-89, and you can administer that. And here's a patient with lymphoma. So you can see the FDG scans that shows you the metabolic the increased metabolic activity, but doesn't really tell you whether this patient has is CD20 positive. But with the zirconium-89 labeled rituximab and imaging over 6 days, you can actually see that this patient was positive and therefore a candidate for rituximab therapy. So a very useful way of selecting patients for a particular therapy. And you can kind of see that the whole body PET would be very useful in these types of experiments. Here's an example using the same labeled antibody of a patient that was a patient that showed lung cancer patient, and you can see FDG uptake in this lung cancer patient. The rituximab PET imaging was also positive, so this was a perfect patient candidate for treatment with rituximab. And 3 months later, FDG showed you don't see the localization in that lung tumor, showing the effectiveness of treatment. So here's an example where you can use PET to select a patient for a particular treatment, but also be able to measure the effectiveness of that treatment. And of course, there was 2 different types of tracers there, the small molecules, where we looked at the pharmacokinetics and pharmacodynamics. And you can do that because with small molecules, kinetics are very fast, and you have different types of imaging protocols, mostly dynamic because of the fast kinetics. With the monoclonal antibodies, you saw that the imaging can take up to days with slow kinetics with much larger molecules like monoclonal antibodies, and really perform static scanning. So just to wrap up, drug applications of that sort with the whole body PET or the total body PET is going to be really, really important in progressing the innovation in drug discovery forward, because you can look at pharmacokinetics of drugs in the whole body at the same time. But importantly, the low dose that's required for total body PET means that you can do these kind of repeated studies that you need to do in drug studies, but also able to study normal biology, which isn't really possible with these kind of repeated studies on conventional PET scanners. So a very welcomed innovation in addition to our imaging capabilities at the university.